Welcome everybody to this EU-IPFF webinar on the genetics of pulmonary fibrosis, which is the second in a series of monthly webinars we're holding leading up to the second European Pulmonary Fibrosis Patient Summit in November 2022, this year. My name is Steve Jones. I'm the president of EU-IPFF, the European Pulmonary Fibrosis Federation, which is a collection of 21 pulmonary fibrosis patient organizations from 15 different countries across Europe. I'm also the chair of Action for Pulmonary Fibrosis, a charity working in the UK. Tonight, it gives me really great pleasure to introduce a friend of mine, Dr. Richard Allen, who is a scientist from the University of Leicester in the UK, who is a leading researcher in Europe on the genetics of pulmonary fibrosis. He's been doing an amazing amount of research in the last few years, largely funded by Action for Pulmonary Fibrosis, which has resulted in a series of really important articles in international journals. We're especially grateful to Richard coming tonight because 4 days ago, he hadn't done his slides and he was still on his honeymoon. So as soon as he got back, he got down to producing the slides and sending them over to us. Richard is going to talk for about 25 minutes. And we'll then have a Q&A session for another 25 minutes or so. We aim to finish tonight by 2000 CET or 1900 UK and Ireland time. Could I please ask you UK, Ireland and Portugal time? So without further ado, it gives me great pleasure to welcome Dr. Richard Allen and to invite him to talk to us about the genetics of pulmonary fibrosis. Richard.
Thank you, Steve. For that introduction. As Steve said, I'm a research fellow funded by Action for Pulmonary Fibrosis, based at the University of Leicester in the UK. And today I'm going to talk about the work that we've been doing looking at the genetics of pulmonary fibrosis. So to start off with, just give a summary of pulmonary fibrosis, and then some introduction as to what DNA is and what genetics is. Then moving on to looking at the genetics of IPF risk, so trying to understand why people get pulmonary fibrosis. Moving on and looking at the genetics of progressive IPF, so why do people's diseases act so differently? Why do some people have a very stable form of the disease, whereas other people have a more progressive form of pulmonary fibrosis? And then finishing with a summary of all the things that I spoke about. So to start off with, just as for what is pulmonary fibrosis, this is sort of a summary for everyone. I'm sure that many of you sort of fully understand what pulmonary fibrosis is. But if we just look at a diagram of a lung, so this is a sort of simplified diagram. We have these two big air sacs. You breathe in air, it comes down through the windpipe, it kind of spreads out into the bronchus and bronchioles, and then it goes to these little air sacs at the end, which are called alveoli. And those are the parts of the lung that are affected during pulmonary fibrosis. So if we zoom into these alveoli, this is sort of a representation of what they look like. So they're these little air sacs shown by these sort of purple cells, and then around the outside is blood vessels. So what happens in a sort of a healthy lung is we have red blood cells come along with carbon dioxide that the body's used. When we breathe in, oxygen gets drawn into these alveoli, and because the wall between the alveoli and the blood vessels are so thin, it means that gas can transfer in and out of the bloodstream. So we can get rid of carbon dioxide from our bloodstream and we can get oxygen into the bloodstream. And then that oxygen is taken away and used throughout the body. On the right, we have a representation of a fibrotic lung. And with this, we believe that there's some initial damage to the lungs for some reason. And then there's, again, for another reason, there's a progressive buildup of scar tissue. So this is shown by this sort of grey area that fills in the space between the air sac and the blood vessel. So what this means is that the lungs become really stiff. So now when red blood cells come along with carbon dioxide, because the lungs are now really stiff, it's much harder to breathe in, and so you can draw much less oxygen into the lungs. The second thing is it also means that this the wall of this air sac is now much thicker, and that also impairs how well the gas can transfer from the blood vessels into the from the blood into the alveoli. And then this scarriness spreads throughout the lungs progressively until the lungs become stiffer and stiffer. There are many risk factors associated with pulmonary fibrosis, and we think that there's sort of an interaction between many of these together. There's not just a single cause that causes you to get pulmonary fibrosis. So particularly sort of inhaling stuff that can damage your lungs, or exposure to dust, mold, smoking. Pulmonary fibrosis tends to affect more elderly people. And we also know that genetics plays quite a key role. And then I'll talk a little bit more about that today, and how genetics can play a role in pulmonary fibrosis. So the obvious question is, so what is DNA and what is genetics? So in essence, DNA is essentially just the instructions that make us who we are. So in every cell of our body, we have this set of instructions that determines sort of what we look like, maybe our risk of certain diseases, but it's sort of a set of instructions that builds us. So everyone's DNA is all different, apart from for identical twins, and it's these sort of differences in our DNA that make the differences between us. So you may have seen sort of a structure like this before, which is this double helix structure, which is what DNA looks like essentially. So it's just a string of chemical bases that follow this double helix pattern. But actually, when we look at DNA, there's only actually 4 chemical bases involved in making this set of instructions. And we can label these. So we label these as A's, C's, T's, and G's. So if we look at this stretch of DNA in this person, we can see that at their first position, they've got an A, at their second position, they've got a C, and then a T, a C, T, and it carries on. And this will carry on throughout the whole DNA. And we have 2 copies of DNA. So we inherit one from our mum and we inherit one from our dad. So we always have 2 copies of our DNA. So if we look at the first position, we can say that this person has an A on both sets of the DNA, and then they've got a C at the next position on both sets, and then a T and on the next set. And for the vast majority of our DNA, we are the same. But you can have places where there's differences. So I'm going to refer to these as genetic variants. So in this example here, this person has a G on the bit of DNA that they've inherited from their mum, and they've got a T from the bit of DNA that they've inherited from their dad. So we would call this person GT in this position. Whereas other people might have a G on both sets, so we'll call those GG, and other people might have a T on both sets, so we call those TT. And then just to give some sort of definitions for words that I might be using, so you may have, I'm sure most people have heard of the word gene. So in your DNA, there are certain parts of it that have sort of a small set of instructions that make something that's called a protein, which is essentially sort of the building blocks that make the body. And where you have these parts of the DNA, this is called a gene. And genetics is essentially just the study of genes and DNA. And the other thing to take into account is that DNA is actually surprisingly huge. So although I've got an example here of a short stretch of DNA, it's actually 3 billion base pairs long. So you can actually write out someone's DNA, and it would be this collection of 3 billion A, C's, T's, and G's. So in terms of how do we study genetics, well, say from the genetic variant on our previous slide, that example, this position where you can either be GG, GT, or TT, we might say, well, does this genetic variant have anything to do with pulmonary fibrosis? So what we would do is we would collect the DNA of lots of people without pulmonary fibrosis and collect the DNA of lots of people with pulmonary fibrosis. And then at this position, we would work out whether they're GGs, whether they're GTs or TTs. And here's a representation, and we can see that if we look at people without pulmonary fibrosis, lots of people are GG and there's a few GT people. Whereas if we look at people with pulmonary fibrosis, we can see that the T base is much more common. And we can see that if you have a T, and the more copies of the T allele you have, the more increased your risk of IPF is. And this is a true example. So there is a position in the DNA where if you have a T, if you have just one copy of a T, you are 5 times more likely to develop IPF than if you had GG. So, which is quite incredible because, as I said, it's 3 billion. This DNA is 3 billion base pairs long, and if you change one G to one T, then that increases your risk of pulmonary fibrosis 5-fold. So, once we identify a genetic variant like this, we can then try and work out, well, why does that increase your risk of IPF or pulmonary fibrosis in general? So in this example, I've chosen a genetic variant that lies near a gene called MUC5B. And what that gene does is it produces mucus in the airways. And what we think is that, well, what happens is people with this genetic variant, they produce more mucus, essentially. So what we think is that this increased level of mucus can impair how well the lungs defend themselves. So you may be more susceptible to damage in your alveoli, and it may also impair how well the lungs can repair themselves. I think one also key takeaway is that these determinants aren't just if you have a genetic risk factor, that doesn't mean you will go on to develop pulmonary fibrosis. In fact, if we look at this example and we look in European populations, about 1 in 5 people have at least one copy of the T at this position. And obviously the vast majority of these people will not go on to develop pulmonary fibrosis. But what we think is that these genetic variants can increase your risk of the disease. And then if you have some maybe environmental exposure, for example, if you inhale dust and you're genetically susceptible to pulmonary fibrosis, then that is what leads to the scarring in the lungs. So what we do for our studies is we don't just look at a single genetic variant, we try and look at as many as we can. So we try and look across the whole DNA, look where we know these genetic variants are and test each one separately to say, is this variant associated with pulmonary fibrosis? And then go to the next variant, is that variant associated with pulmonary fibrosis? And so on until we've looked across the whole DNA. And these are called genome-wide association studies, also sometimes referred to as GWAS. So this is obviously computationally intensive. So I know that sort of when you think of how science works and what science looks like, your sort of mind immediately goes to people in sort of white lab coats with pipettes. But actually for us, it's more computational. So it's more using computers. Obviously, if you've got copies of DNA of thousands and thousands of people, these files are huge. So a sort of a normal computer wouldn't really be able to deal with that sort of size of files. So on the right here is a picture of the file system that we use, which is a sort of a supercomputer that's at the University of Leicester, and that anyone at the University of Leicester can use. So what we do is so all the data is stored on this supercomputer, which is called Alice. We write some computer code, send that off to Alice with this code, and the code will tell the computer how to analyze this genetic data, and then we get the results sent back to us. So the benefit of this is that we can actually access this computer from anywhere. So although we've all been working from home mostly for the last 2 years, because we can access this remotely, we've actually been able to continue with the research into genetics of pulmonary fibrosis fairly sort of unhindered. So now we know sort of how you would study the genetics. The obvious question is sort of, well, what's the point? Why are we doing this? So the first point is, if we can figure out parts of the DNA that are causing pulmonary fibrosis, we can then figure out what those genetic variants are doing. So we've already shown that with the MUC5B and the mucus example, where we identify a genetic variant associated with pulmonary fibrosis. We work out what that genetic variant does, and that highlights a sort of a biological mechanism that's important in the disease. So in that case, mucus production. And if we know what's causing pulmonary fibrosis, we can then try and develop treatments that target those processes, and hopefully develop sort of new treatments. The second thing is that we may also identify other traits that have a similar genetic makeup to pulmonary fibrosis. So we may do these studies and we identify a certain part of the DNA that's possibly causing the disease. And if we know that there's other traits that are also caused by the same part of DNA, and those traits already have a treatment, then that may highlight a new treatment that we can then use in pulmonary fibrosis. So this is known as drug repurposing. And finally, we may also find that if we look within pulmonary fibrosis and try and find groups of people within pulmonary fibrosis that have certain sort of genetic makeups, we may find subsets of patients that have a different sort of genetic profile. And in that case, we may find that certain treatments are better for certain people based on their genetics. And then that way we can try and develop treatments that target each person separately, depending on what their sort of genetic makeup is and what sort of underlying biological mechanisms are causing pulmonary fibrosis in those people. And you may have heard the term personalized medicine, which is what this is, where essentially we're trying to develop treatments that target each person specifically. So from now on, I will talk about mainly idiopathic pulmonary fibrosis. So the reason for this is that for these studies, we need lots and lots of people, and as idiopathic pulmonary fibrosis is one of the more common forms of pulmonary fibrosis, we've tended to study that most. However, there has been recent evidence that suggests that drugs developed for IPF are also effective for other forms of pulmonary fibrosis. And actually, there might be quite similar genetics between idiopathic pulmonary fibrosis and other forms of progressive pulmonary fibrosis. So although I will be talking a lot about idiopathic pulmonary fibrosis, the results should also be relevant to other forms of pulmonary fibrosis. So to start with, I'll talk about the work that we've done looking at the genetics of IPF risk. So as I said, trying to understand why do people get IPF in the first place. So to do this, we've done these genome-wide association studies. So we had 5 different studies that we were able to perform these GWAS in, so these genome-wide studies. We had 5 different studies from across the UK, the US, and Spain. We ran these studies, so tested each genetic variant separately to see if it was associated with why people were getting IPF. And then we were able to combine these together into one large analysis that we call a meta-analysis. So this schematic gives an idea of the sort of sample sizes. And so in these, we tend to refer to people with a trait as cases. So people with IPF as cases in this case, and then people without IPF as controls. So in the end, we were able to investigate 7.5 million genetic variants across just over 4,000 people with IPF and 20,000 people without IPF. And these are how we normally present the results. So on this, each dot is a single genetic variant. So if you zoom in, you'd be able to see that there was 7.5 million dots on this plot. As we move from left to right, that's just where it is in the DNA, essentially. As I said, we can write these base pairs all out in one long line. And as we move across, it's as you move along the DNA, essentially. And the higher up the dot is, essentially, that's how likely that variant is to be associated with disease risk. So what this means is that where we see these peaks, so these straight lines, these are parts of the DNA that we think have some role in causing IPF. And I've labeled these with the genes that these peaks implicate. And we can see here that we have the MUC5B gene labeled on one of these peaks that I described earlier. So now we know what types of genes are involved and which genes are involved in why people get IPF. We can then try and figure out what do those genes do. And they tend to sort of fall into certain categories. So some of those genes are involved in lung defense. So for example, the mucus example that I gave earlier. So the idea that if you've got a genetic variant that maybe makes your lung more susceptible to damage in the first place, you may be more at risk of pulmonary fibrosis. Other genes are involved in how well cells stick together. So in your body, you're made up of these tiny sort of building blocks called cells, and they stick together with this sort of sticky stuff essentially between the cells. And if that's broken or cells aren't as sticky, then that may increase your risk of IPF. In our body, our cells are constantly regenerating. So they do this through something called cell replication. and we find that genetic variants that maybe cause issues during this cell replication, they can also increase your risk of pulmonary fibrosis. We can also measure sort of the age of cells. And what we find is that genetic variants that increase how quickly your cells age are associated with pulmonary fibrosis. And this may also explain why pulmonary fibrosis tends to affect more elderly individuals. And finally, there's this process called fibrotic signaling. And I'll go into a little bit more detail as to what this is. So if we assume that this is sort of a picture of a piece of the lung, and we've got all these different lung cells shown in pink, and in the middle we've got a damaged lung cell. So in the body, there are certain proteins that go around and they identify where there is this sort of damage, and they tell the body to repair it. And the body repairs it by depositing scar tissue. So it's much like putting a plaster over a wound. These proteins go around and say, there's this part of the lung that needs repairing, put some plasters over it, put some scar tissue over it to sort of fill that hole. And once that damage has been repaired, there are other proteins that come along and tell the body to stop depositing the scar tissue. So this is a sort of normal process, and this is what should happen. We can almost think of these sort of green proteins that tell the body to deposit scar tissue as sort of accelerators, and we can think of these red proteins that tell the body to stop as brakes. So if we look at one of the genes that we've identified in our studies, it's called ACAT13. And what we found is that the genetic variant associated with disease risk causes you to produce more of these accelerator proteins. So what that means is that although you have these proteins telling the body to stop, because you've got lots of these accelerator proteins, we believe that that means that the body just keeps depositing the scar tissue. And then to look at another example of one of the genes we've identified is called DEPTOR. So if we go back to the example of, we have this part of damaged lung, we have proteins that come along, tell the body that this part of the lung needs to be repaired. So the body will deposit the scar tissue. But what DEPTOR does is it knocks out and stops these brake proteins. So it's almost like it cuts the brakes. So now there's nothing telling the body to stop depositing scar tissue. So the body will just carry on depositing this scar tissue, which is why we think that the scarring progresses throughout the lungs. The reason these are interesting is that there are studies at the moment looking at possible drug targets in both of these accelerator and these brake proteins. This is showing sort of two parts of biology that the drug targets trying to be developed that hopefully will be effective in treating pulmonary fibrosis. So just to give a summary of the genetic IPF risk, by comparing the DNA of people with and without pulmonary fibrosis, we can identify bits of biology that are causing or are important in the development of pulmonary fibrosis. And if we know what's causing pulmonary fibrosis, hopefully we can develop treatments that target those processes. And the other thing to take away is that there's lots and lots of different parts of the DNA that affect someone's risk of IPF. So it isn't just a case of if you've got a single genetic change, you will get pulmonary fibrosis. And it's also, if you don't have a certain genetic change, it doesn't mean you won't get pulmonary fibrosis. There's lots of different factors that affect our risk, including lots of different genetic factors. So to present some really new work and really new exciting work, we also have done these genetic studies to try and identify parts of the DNA that cause a disease to progress either quicker or more slowly. So I'm sure many of you have experienced doing sort of some of these lung function tests. So on the left here, we have an image of someone doing spirometry. So that's where you breathe, take a big breath, and then you blow as hard as you can into a tube for as long as you can. And one of the measures we can get from this is called FVC. So that essentially gives us a measure of how much air that your lungs can hold. You can also do a similar test, which measures, and we get a measure of something called DLCO, which is a measure of how well oxygen and carbon dioxide can transfer between the bloodstream and the air sacs. And if you remember what I was saying at the start, so both lung capacity and gas transfer are decreased when you develop pulmonary fibrosis. And over time, as the disease progresses, your lung capacity and gas transfer decrease. So if we wanted to track someone's disease course, what we would do is we would, when they were diagnosed, maybe ask them to do a spirometry. So say we've got two people, and this is a representation of what their lung capacity is at time of diagnosis. And then maybe a year later, we'll ask them back and ask them to do another spirometry and measure their lung capacity and see if it's changed or not. And we can carry on doing this over a number of years. And we can see that person one, although their lung capacity is decreasing slowly, it's a relatively stable form of the disease where the lung capacity hasn't drastically reduced. Whereas person two, we can see that their lung capacity is quite progressively decreasing each year. And then what we've done is we've done a genome-wide association study to test, are different genetic variants affecting how quickly your lung capacity or your gas transfer decrease. So to take away the sort of key findings of this, we identified a genetic variant that's associated with changes in how quickly your lung capacity decreases. And this implicates a gene called PKN2. So we've got this genetic variant that we think is somehow linked to how quickly your lung capacity decreases after you've been diagnosed with pulmonary fibrosis. And the reason this is sort of really exciting is that currently in cancer studies, there are drugs being developed that target PKN2. So this is highlighting sort of another potential drug target that could be potentially used in pulmonary fibrosis. And the other interesting point is that, and it's also been quite surprising, is that the genetics of disease risk are quite different to those of progression. So the genes that we identified in the studies that I presented earlier for sort of why do people get pulmonary fibrosis seem to be very different to the genes that come up when we look at disease progression, which could have a huge impact in terms of sort of drug development, because you obviously, if you're developing drugs, probably drugs that target processes that are causing the disease to progress are probably going to be more effective than developing drugs that target processes that cause you to get pulmonary fibrosis in the first place. So just to give a sort of summary of the whole presentation, there are lots of parts of the DNA that have an impact on whether you get pulmonary fibrosis or not. The other key thing is that just because you have a genetic risk factor, that doesn't mean you will get pulmonary fibrosis. So I do get asked a lot of questions about, I have pulmonary fibrosis, what does that mean for my kids or my grandkids? So if you have a genetic risk factor, then you may pass that on to your kids, and that may increase their risk of pulmonary fibrosis, but it definitely doesn't mean that they will get pulmonary fibrosis. If we think back to that example at the start, where 1 in 5 people carry this genetic risk factor, which has a huge risk on a huge impact on disease risk. So just because you have a genetic risk factor, that doesn't mean that you will get pulmonary fibrosis. And the other thing to take away is that there is lots of research that is happening that's hopefully moving us closer to developing new and effective treatments. Another question I get asked quite a lot is, how do I get involved in research? So the best place to go to is to ask your doctor to see if they're recruiting for fibrosis research. If they say they're not, then ask them why not, because I think that research is important, and the more people involved, the more we can study. In terms of sort of resources for finding where studies are recruiting people, in the UK there's clinicaltrials.gov, which has a link for all clinical trials that are ongoing, and there's also the EU-IPFF clinical trial finder. just to finish off with some acknowledgements, there are lots and lots of people involved in these studies, but particularly thanks to Louise and Ray, Justin, Carlos, Gisli, Shrufan, Phil and Brian, who played a key role in these studies. Thank you to the funders. As Steve said at the start, I'm funded solely by Action for Pulmonary Fibrosis, which is a patient-led charity. And mainly thank you to everyone who's agreed to take part in these studies, as we definitely wouldn't be able to do any of this research without them. And thank you everyone for listening. If you have any questions, then I'll be happy to answer.
Thank you very much for a really stimulating chat. No, I think that was great, and we've got a number of really interesting questions coming up. I mean, one of the first ones is actually the very question you just answered, but maybe you'd like to have a go at it again. You know, my husband died aged 58, having had IPF for about 6 years. His mother died age 76 with IPF, as did her brother. How can I look at the genetic risk to my son for his future?
Yeah, so as I said, sort of, we do see this sort of pattern of pulmonary fibrosis in families because there are these genetic risk factors that can get passed down between different generations. And obviously, people we're related to, we share more of our genetics with. So if someone has a genetic risk factor, then that can get passed down different generations. I think one of the things to stress is that, as I said, just because you have a genetic risk factor, that doesn't mean you will get the disease. We're not really in a position at the moment to sort of predict who will develop pulmonary fibrosis and who won't. So in terms of genetic testing, you wouldn't get sort of a definite answer for yes or no, but I know that there are some medical centres, I think, do look at genetics to try and understand someone's disease risk.
Yeah, that's I must say, one thing I should mention is that we're doing this webinar tonight in order to feed into a major session that we're going to have at the summit, when we're going to have doctors from different European countries talking about the whole issues of genetic counselling, whether to do it or not. You know, there are lots of questions, as Richard said. You don't really know what the gene, if you have familial in IPF or in your family there's IPF or familial pulmonary fibrosis, you don't really know what the genetic changes that's happened, and it becomes very difficult to trace it through, and counselling becomes really difficult except in certain circumstances. I think, as Richard, I'm right in saying that as a result of your work and others, we're getting now a much better understanding of the genes to look for, but it's still not perfect.
Yeah, and I think the MUC5B example probably shows it perfectly, that we've got this genetic variant that massively increases your risk of pulmonary fibrosis. We don't really see that with any of the other genetic variants, but even that's not a very good predictor of whether someone will develop pulmonary fibrosis or not. And even most people who have that genetic risk factor, they will not go on to develop pulmonary fibrosis.
Yeah, somebody's asked a specific question on MUC5B actually, saying, does your study show if the variant appears more in sporadic IPF than it does in familial pulmonary fibrosis?
Yeah, so yeah, I probably should have said that. So mainly our research has been based on sporadic IPF. So we haven't really looked at familial IPF. You do get certain genetic changes that come up in familial IPF studies that don't come up in the sporadic research. So for example, if you look into the literature, there's things called like surfactant proteins, and maybe stuff related to telomeres, which is that biological aging process. But there is also quite a lot of overlap in the genes that come up when you look at familial fibrosis and IPF.
Great. Okay. I mean, there was a question in the chat. Please put your questions in the Q&A, as most of you are. But the question in the chat was, what percentage of pulmonary fibrosis patients have familial? And I know there's quite a debate on this, but most people I've said say 15 to 20%. Is that?
Yeah, if I'm honest, I'm not entirely sure what the exact figure is. It sort of becomes really difficult in terms of sort of trying to estimate these and trying to like find family history, and a lot of people may not know their sort of family history. So but yeah, I don't know the exact figure.
Yeah, I'm like that. I have one side of my family, they all live till they're 90. The other side all died before they were 60 of heart attacks and other things. I don't know whether my pulmonary fibrosis, I had IPF before my transplant. I don't know whether that is familial or not, because half my family's not there anymore. So it is a problem, isn't it?
Yeah, no, it's actually surprisingly hard to sort of estimate those sort of figures.
On the question of telomeres, there's an interesting question here. Have you seen evidence, as with Colleen van Morsel's work in Holland, of family members with short telomere syndrome who do not have the known telomere mutation of their relatives with pulmonary fibrosis, and that yet they still go on to develop IPF or PF?
Yeah, I don't really know that research. Sorry, I'll look at the.
Yeah, it's the very first question.
Yeah, so no, essentially we haven't sort of looked at that kind of thing. When we look at sort of familial PF, what you tend to find is that you have these sort of genetic variants that are quite rare, so they tend to just be in certain families. When we do the studies that we do into sporadic pulmonary fibrosis, what we tend to look at is these genetic variants that are quite common in the population. So a lot of the work we've done has been looking at these sort of genetic variants that are quite common in when you look in sort of general population, as to there's lots of people that have these genetic variants and how they can change your risk of either disease or how progressive the disease is. But looking at sort of specific rare genetic variants, we haven't done that as much at Leicester.
Right, and I guess over time we'll find more and more of that work will be done as these genetic variations are identified. Yeah.
Yeah, definitely. I mean, one of the issues is cost, because you can measure these sort of common genetic variants relatively cheaply in lots of people. But if you want to try and find these rare genetic variants where you might not necessarily know where they are, you have to essentially read the whole DNA. And as I said, it's 3 billion base pairs long. So to measure that whole DNA is really expensive, and that can limit sort of the sort of study sizes. But when we look at these rarer genetic variants, which is another reason to sort of why we've at the first point started to look at these common genetic changes, and then as we develop bigger data sets with maybe more detailed DNA data, we can start looking at these rarer changes, which we know they do have some effect on disease risk.
Yeah, I mean, the rate of change is fantastic at the moment, isn't it? I mean, 5 years ago, we knew very little. And now, I mean, say you the story, when you started your PhD, how many variations? When I didn't know, I had 17, wasn't it? Or
yeah, I think
associated.
Probably about 10.
10 were identified as associated with IPF.
Yes.
And how many do you have now?
So I think we're up to 2020 now. So we've doubled them in the last couple of years, and then plus we've also got now the progression studies that we've started doing. So we've identified, we've almost sort of doubled our understanding of the genes involved in pulmonary fibrosis in sort of the last couple of years.
Yeah, and as the understanding of genetics in general speeds up, so will research relating to IPF speed up. Yeah.
Yeah, and as the technology as well, sort of for how you measure DNA improves, that'll also help us.
An interesting question here. Any research on ethnicity, the presence of risk of T factor in ethnic groups? Do we know whether that exists or not?
So yes, that's a really good question. I'll use the MUC5B example again, because that's a really perfect example in this case. So when I say that 1 in 5 Europeans have the copy of this T allele, this T at this position, if you look at different ethnicity groups, you actually see it's way less common. So there's only a few people, if you look in sort of South Asian or East Asian populations, there's only a few people who will have this T at this position. And if you look at African populations, then practically no one has a T at this position. Just sort of how, when sort of humans spread around the world, you get certain mutations that sort of appear, and then they tend to cluster in certain areas. But this mutation is clustered in Europeans. But what it does is it actually matches the prevalence of IPF. So where you see IPF is most common in, so IPF is most common in European populations. It's slightly less common in sort of South Asian and East Asian populations, and it's fairly rare in African populations, which matches how common this T is in the different populations. So where we see populations where this T is quite high, we see higher levels of IPF, and where this T practically doesn't exist in populations, we don't see as much pulmonary fibrosis. So there is massive differences, well, not massive differences, but there are differences in different ethnicity groups. So far, we have focused on European populations just because that's where the large data sets have been collected. But we are just starting now on looking at the effect of these variants in different ethnicity groups to see how generalizable are our results and to make sure that our research is represents everyone.
Yeah, I mean, we know generally, don't we? I mean, IPF is 70 plus year old people, 2/3 men, 1/3 women, mostly, almost totally European. Yeah, so it's a old white man's disease. That's why I got it. Okay, but we also know that the other forms of pulmonary fibrosis, like rheumatoid arthritis, ILD, scleroderma, hypersensitivity pneumonitis, all subtypes of pulmonary fibrosis that the patient experiences in almost exactly the same way as an IPF patient experiences their disease. They tend to affect younger populations, age 40 to 60, and there are far more ethnic diversity and far more women. In fact, marginally more women in that group than men. So do we know the genetic underpinnings or reasons for that?
No, so yeah, so we don't yet, essentially. So we're doing some work here in Leicester, specifically looking at hypersensitivity pneumonitis. So one of the benefits of being Leicester is that we've got quite a diverse population, particularly sort of a South Asian community where hypersensitivity pneumonitis is slightly more common. So we're sort of looking more into the genetics of hypersensitivity pneumonitis, but we haven't done that work yet. So in terms of looking at sort of genetics across those different diseases, we don't understand those diseases quite as well as we understand IPF.
No, yeah, I've been talking to an Indian doctor recently, with a view to forming support groups in India for patients, and he was saying the majority of the diagnoses they have are actually hypersensitivity pneumonitis, not IPF. But again, it's a difficult disease to diagnose as well. So, but an interesting question here, which I don't think you'll be able to answer. And Becky, who's asked it, I think this is one to bring to the summit for our discussion about new drugs and where the pipeline is going. Is hi, other than PKN2 targeting drug used for cancer, how far are we in the world of medicine in terms of coming up with medications that stop disease progression rather than just slowing it down? And has a PKN2 cancer drug been tried on IPF patients yet in real life? And for me, this is the absolute fundamental question. I mean, we all talk about wanting to find a cure for pulmonary fibrosis. What a cure means is the way Becky's defined it there. It is that we stop progression. Like HIV/AIDS, there's not a cure for HIV/AIDS, but we found a way with antiretrovirals to actually stop the disease. So I would hope that if in 20 years' time, when patients present to a doctor, I hope he or she would be able to say, you've lost 10% of your lung function. Sorry about that. Take these pills and you'll live a long life, and you'll die of something else. And hopefully he'll be able to say we can resuscitate your lung, but that's a different question. But yeah, sorry, I talk too much. Your turn, Richard.
No, yeah, I don't really have much to add. Yeah, it's, as you say, at the moment, the drugs just slow down how fast the disease progresses. If we can identify what's causing the scarring to spread, and we can develop treatments that stop those processes, then hopefully we'd be able to stop the scarring spreading even more. Whether that leads to a cure, not sure, but hopefully, as Steve says, we can at least stop the disease progressing any further. And as far as I'm aware, I don't think the PKN2 drug has been used in IPF patients yet. This is sort of really new work that we've sort of only just, it's not even published yet. It's still going through the sort of peer review system, so it's really new stuff. We haven't got to the later sort of stages of research to test whether these drugs can be used in IPF patients. Yeah,
and another very interesting question again. One of the people attending says, I was diagnosed with RA-ILD, rheumatoid arthritis ILD, 4 years ago, and then diagnosed with cirrhosis of the liver last September. Does this mean I have a higher risk of developing scarring in other organs in my body?
Yeah, so I'll probably focus more on the sort of genetic side of that, of the answer. I think that there are certainly you certainly see between liver and lung. I think some people with lung fibrosis then do go on to develop liver fibrosis. In January, we just started a big project called Demystify that I know at least Action for Pulmonary Fibrosis are involved with, and I don't know if any of the other support groups are involved with, which is essentially trying to answer this question. So, so looking at fibrotic multimorbidity is called, so where you get fibrosis in different organs and trying to understand why some people go on to develop fibrosis in multiple organs. So is there a single sort of fibrotic mechanism that causes fibrosis in lots of different organs, or are the different organs quite distinct where you have a lung fibrosis mechanism? So for example, we could think of the mucus example is probably mainly going to affect parts of the body that develop mucus, so particularly sort of the lungs. But as of yet, we don't fully understand the reasons why some people will develop scarring in multiple tissues. But hopefully over the next couple of years, we'll massively increase our understanding of that. Yeah, and understand different maybe sort of clusters as to certain organs that have similar makeup in terms of genetics.
Yeah. No, thanks, Richard. There are two interesting links being provided in the question Q&A section. I don't know if all participants can see all questions or not. Alba, what do you know?
No, they can't see it, but I can publish it in the chat.
Okay, we will publish these two links from the chat. One from DC Action, working with families with short telomere for a particular genetic disease, and another one from the Team Telomere has just published their clinical guidelines, which has a chapter on pulmonary fibrosis. So both of those will be provided to all participants after the event. Another question, again, following a similar course, is there any commonality between the genetics of people with multiple sclerosis? Because this person says 3 members of my family have had, have had multiple sclerosis, and IPF, I think. Anyway, is there an association between IPF and multiple sclerosis? She says, we'd love to participate in your research, and goes on to say, why are GPs and other doc consultants obsessed with acid reflux, which I don't have? And I agree, I didn't have acid reflux either, and I had IPF. But the question really is about multiple sclerosis and IPF.
Yeah, so not that I know of, certainly not the genetics. So none of the genetic variants that we've found have been reported as being associated with MS. So as far as I'm aware, there isn't necessarily a genetic link between those. It may be that in your family you have people who have carry sort of MS genetic risk factors and unfortunately carry IPF genetic risk factors as well, that are both being sort of passed on separately. But as far as I'm aware, there isn't a genetic overlap between the two traits.
Okay, thank you, Richard. And what might be our last or one of the last questions, if a genetic, there's one question actually about nintedanib damaging my liver. You know who you are. I mean, if you'd like to send an email in, you can send it to steve.jones@eu-IPFF.org. Send that question in and I'll get it answered for you by a medical doctor, the second part of your question. Okay, and then if a genetic or familial connection of IPF is confirmed between two close relatives, what are the chances that both will have a more or less identical development of their PF disease? In other words, is there a chance that one of the relatives may have a much slower or faster development than the other?
Yeah, so that isn't sort of something we've looked at specifically, but I think what our research has shown, which as I said was actually really surprising, is that the genetics of disease risk aren't necessarily the same of genetics of disease progression. So if maybe there's been a genetic variant that has caused both individuals to develop pulmonary fibrosis, but they have these other, but they're sort of different in these other progressive genes, if we call them that, I think it's quite likely that the disease will progress quite differently in two different people.
Okay, thanks. There's a question here which says, which I could quickly answer. So would you separate UIP, a UIP pattern, as a separate genetic illness to IPF? And the answer is no. UIP is one of the defining characteristics of a diagnosis of IPF. It's a little bit wider than that, but until you're pretty clear that it's a strongly UIPF or an indicative of UIP pattern, you will not be diagnosed with IPF. And in fact, I was not diagnosed with IPF for 5 years, I think, until finally the pattern showed well enough that they said, this is IPF that you've got. And the final question, which is an interesting one, is if IPF is genetic in origin, why does a lung transplant stop it from restarting?
Yeah, so I suppose one of the answers is that if you have a new lung put in, then the cells in that lung will have a different genetic makeup. So they will have the DNA of the person that you got the lung from. I think that your genetics can affect possibly how well the lung takes. So if you look at the sort of telomere gene, so this sort of biological aging, I think that can affect how successful the transplant is. But I think if it is a successful transplant, then you've essentially got a new lung in that has a different genetic makeup, which may explain why the new lung isn't affected by pulmonary fibrosis like your original lungs were.
Right. I think that's right. I think if there were a propensity to developing IPF, it wouldn't probably, most lung transplants, sadly, I mean, it's generally 5 to 6 years after transplant is the average life expectancy. And so it wouldn't have had time to develop as IPF. If mine went on for 20 years, hopefully, maybe I might sadly develop IPF at the end of that time, because it takes time to come through properly. Great. Well, Richard, that is wonderful. Thank you very much for a really stimulating and great session. I just reiterate again for everybody listening that we are going to have a session in the summit. The summit is on the 3rd to the 5th of November. And you'll be hearing a lot about this in our media in the near future. And there'll be a session there on the genetics of IPF, where we're looking at these questions of familial IPF, genetic counselling, what should we do about it across Europe? And whereas we've had the science tonight with Richard, the genetic science, we'll have the clinical aspects of how you deal with this in a clinical situation then. It's been a great session tonight. Thank you again very much, Richard. Thank you all for coming and for asking your questions and taking part in the discussion. And thanks very much to Alba and the team at Patvicates who run these webinars for us so faultlessly. So Richard, once again, thank you very much indeed. It's been great to see you tonight, and look forward to continuing to be associated with you. So thanks very much, everybody.